Hello everybody, this is Akshay over here and welcome to Akshay's Biology classes. So like every year in the month of October, the Nobel Prize is awarded. Likewise, on the October 5th of this year, the Nobel Prize for Physiology or Medicine was also awarded. And this prize was mainly shared by three people, Harvey Alter, Michael Houghton and Charles M. Rice, for their discovery of Hepatitis C virus. So in this video, we will mainly try to understand their work and why their work deserves a Nobel Prize. The story actually begins with Mark Bloomberg, who got Nobel Prize in 1976 for his discovery Hepatitis B virus, that is HBV. And he discovered this virus by detecting a antigen during his routine serological tests. With further tests, he could conclude that the antigen is actually part of a virus and that virus is hepatitis B. So he could develop highly sensitive serological tests which could identify the carriers for HPV. As a result, the post-blood transfusion hepatitis B cases could be reduced. So this finally reduced the post-blood transfusion hepatitis cases. Although it reduced only to 20%, the remaining 80% of the post-blood transfusion hepatitis cases were still coming into observation. And interestingly, all of these post-blood transfusion hepatitis cases were completely unrelated to HBV. So this led to hunt for the agent that was mainly responsible for this. Here is when, by this time, Harvey Alter, who was working in NIH, USA, studied some of these post-blood transfusion hepatitis cases. And he concluded that with his studies, these new cases were mainly because of a new virus. So in order to identify the new virus, Harvey Alter collaborated with two people, that is Stephen Feinstein and Robert Purcell. So remember one thing, interestingly, Stephen Feinstein and Robert Purcell are the people who identified hepatitis A virus in 1973. So by collaborating with these two people, Alter observed that in the post-transfusion hepatitis cases, he detected an antigen which was neither related to the hepatitis A or hepatitis B virus. And he named this as non-A, non-B hepatitis, abbreviated as NA-NBH. And interestingly, Harvey Alter also observed that when the plasma of the patients who were infected with the non-A, non-B hepatitis were transferred to chimps, even the chimpanzees also developed the symptoms of hepatitis. Now this was an interesting observation because no other mammalian model showed the symptoms for hepatitis. Now this proved that apart from humans, chimps are the only mammals which show hepatitis. Hence, chimps can be used as animal model. And using chimpanzee as animal model, Harvey Alter could characterize the virus and also observe the changes in the hepatocytes of the chimpanzee's liver. And based on these, he published that the virus is mainly having a viral coat, and this viral coat is made up of lipids. And the virus also had a diameter of 30 to 60 nanometers. But however, the identity of this particular virus still remained obscure. And here's where Michael Houghton comes into the picture. Michael Houghton mainly took molecular approaches. So he screened cDNA fragments, that is the complementary DNA fragments, which were isolated from the infected chimps. But however, the problem was these cDNA fragments mainly contained the genome of host also. So to overcome this challenge, Michael Houghton mainly isolated the RNA from the chimps and he prepared a cDNA library. And this cDNA was actually transferred to a high efficient lambda phage system. This bacteriophage system was used to infect the bacteria. So after transferring the cDNA into the bacteria, that cDNA will be expressed in the bacteria. Hence, finally, the bacteria also will show viral antigens. And he cultured these bacterial colonies and screened 1 million colony. But out of 1 million colony, only one colony turned out to have no host sequences. So he used this one colony which was not having any host sequences, and the DNA was used to hybridize with the RNA, and there was hybridization also. With this, he identified the new virus, that is the hepatitis C. But however, there was still a problem. Though Michael Houghton identified this new virus, it could not prove that hepatitis C virus alone causes the post-blood transfusion hepatitis. And also, there are no roles played by the cofactors. This challenge was mainly solved by Charles Rice. Charles Rice looked into the RNA genome of the virus and predicted that the 3' end of the viral genome may play an important role in the infection. So he constructed certain RNA genomes which had this conserved 3' end, and such viruses with the constructed RNA genomes was mainly used to infect the chimpanzee's liver. But however, there was no infection at all. So Charles Rice predicted that the replication mechanism in the case of the virus is very much erroneous. Hence, it may contain some inactivation mutation. So Rice engineered certain set of RNA genomes by removing the inactivating mutations. And such virus was mainly used to infect the chimps. And the result was there were hepatitis C symptoms. With this, Charles Rice could prove that hepatitis C alone causes the chronic hepatitis. So finally, the question is whether this discovery, their painstaking work on the discovery of hepatitis C virus, deserves a Nobel Prize. The answer is yes, indeed it deserves a Nobel Prize. Because globally, around 71 million people are affected of chronic hepatitis C virus. And in 2016, approximately 4 lakh people died because of hepatitis C virus, which had led to cirrhosis and liver cancer. Because of their work, antiviral medicines actually have been developed, and these medicines can cure 95% of the persons with hepatitis C virus in the early stage itself, and even in the later stage also can be cured. Although we don't have any effective vaccine as of now, but still the research is going on to develop an effective vaccine against hepatitis C. As far as our country is concerned, around 40 million people are affected with hepatitis B virus, and 6 to 12 people are affected by chronic hepatitis C virus. Shortly, I will meet you people with another video discussing something interesting which will enhance our understanding regarding biology. Thank you.